Okay, thank you everybody for joining our webinar with today's episode, Landscapes of the Human Mind. This is already the 16th webinar that we are offering from the Human Brain Project, organized in this flagship project of the European Commission, but it's really our idea to give you, everybody, a broad overview of what is happening in research. And today we are focusing on the landscapes of the human mind. And as you can see, we have an exciting program put together, hopefully for you exciting as well. And I will introduce the speakers in a second. But let me start with the notion that the brain is, of course, not only one of the most fascinating systems that we can approach in science, but it's also one of the most complex systems. And this complexity can be seen, for example, in the way how the brain is organized on different spatial scales, but also on different temporal scales. That means that when we are looking to brain organization, then we can do it from the level of single molecules at the scale of certain angstroms or nanometers. And molecules are, for example, involved in neurotransmission, and receptors are important molecules that bind neurotransmitters that transmit information from one cell to the other at synapses at the level of a very few micrometers. Synapses connect neurons, and sometimes these neurons have very, very long branches, axons, several centimeters long. And they connect cells in far distant regions in the brain. Cells form networks with a very complex architecture. This is an example of the hippocampus. And the architecture, of course, has to do something with the function of a certain brain structure, like here the hippocampus. And the hippocampus is only one part of many structures that we have in the human brain. Finally, we should be aware that, of course, the brain is not acting by itself. The brain is part of a body. We as human beings have a body which is extremely important for the way how we are perceiving information and processing information, and how we are communicating with the outside world. So this is what we would like to understand in human brain research. And the question is how to link the many different aspects of brain organization to each other. And one of the answers is, well, let's do it in an atlas. And we know atlases for several centuries, but probably we need some kind of new atlas. And sometimes we compare our human brain atlas that we are developing in the Human Brain Project with Google Maps, because there's a browser, there's a web-based access, there are different options to zoom in and zoom out. But I feel that the atlas approach for the human brain goes beyond that and poses questions to us that are very specific and that are also very complex. So one of the aspects why I feel that it's so complex, this is intersubject variability in brain organization. Brains differ in their size and shape as human beings differ with respect to height or color of their eyes or color of hairs and so on and so forth. And this is an example of intersubject variability of two areas in the visual cortex, areas 17 and 18 and 10 human brains. And the red and green areas are always the same. And you can see that these brain areas vary between the brains in shape, size, also position, but also the size, that is a folding pattern of this region, varies between the brains. And the relationship between borders and size varies as well. And I can add that intersubject variability can also found at the level of the microstructure. And when we started more than 25 years ago to map the human brain, we introduced an algorithm that helps us to identify borders between cytoarchitectonic areas in the brain using image analysis and statistical tools. Here you see such an example that we map cell body stained sections here again in the visual cortex. And using these quantitative tools, we are able to identify borders between brain areas in a reproducible way. And in order to capture intersubject variability, we are mapping not one brain, but 10 brains, and bring this in a so-called reference space to make these cytoarchitectonic areas available as an anatomical reference for data coming from functional imaging studies, for example. The methodology we have published 2.5 years ago and describes the details and the workflows that lead to this atlas. How can it be used? Well, this is one example of many. So you see here on the right top, 3 areas in the premotor cortex. And in the middle, you find again these 3 areas, which are called 6R1, 6V1, and V2. And the little spheres, they indicate clusters of activation of functional imaging studies coming from quite different functional imaging tasks, for example, fulfilling a certain hand movement or having a grasping movement or orient yourself in the space or observing a movement. And we see that there's a certain dissociation between the areas in the 3D space. And this is a possibility to link data and findings from the living human subject that we can measure using neuroimaging with the high resolution post-mortem maps that we have acquired in the past years. And this atlas now has grown and does not only provide cytoarchitectonic maps, as we can see here, but also a lot of tools to get access to other data that can be found for each of the areas. And Timo Dickscheid later will show some applications. So there are different ways to look to the brain, to zoom in, but also to get access to different types of data, to get description of these data, to learn more where it has been published, and to better understand, for example, how a certain brain area is related and connected to another brain area. And we are going also one step towards higher spatial resolution, not only to large systems involved in a certain function like for example, grasping or reaching, but we also wanted to go into more microscopical detail. And what you see here are images obtained with polarized light imaging. This is a technique that allows to image axons, that is the myelin sheets surrounding axons using an optical technique. And in the same images, we have stained and labeled different types of cells, here PAF-albumin, PAF-calbindin, and calretinin cells. And the cell-specific parameters makes it also possible to link this type of atlas data to atlas data coming, for example, from the US Brain Initiative or coming from the work in which Katie is involved. So the idea today is to show you what are the tools to approach atlas data in the human brain. This is done by Timo Dickscheid. And then Tim Salditt will join us. He is a physicist and coming from a different perspective, analyzing microscopical details of tissue structure that are below even the 1 micrometer range and that could benefit from the integration into a larger spatial framework. We will have today here Katie Berner from the US, who speaks about the perspective of the human body. And I'm really excited today also to have her here, because as neuroscientists, we sometimes forget that's not only the brain, but the body is equally important. And at the end, Christian Büchel will close our journey to the landscapes of the human mind. He's an expert in functional imaging and will speak more about from brain structure to cognition and then going back. So having said this, I'm happy now to introduce and hand over to Timo Dickscheid, who's a professor of informatics at Düsseldorf University and working at the Research Center Jülich, and he is the head behind the Atlas technology. Timo, please.
Thanks a lot, Katrin. I was muted for introducing me. Happy to present the infrastructure more or less in a nutshell. What we are doing in EBRAINS in terms of the atlases is to provide infrastructure and tools and services to access atlases of different species. You see here some screenshots of a human brain atlas, a rat brain atlas, mouse brain atlas, and a monkey atlas, which is coming up. And of course, in this webinar, I will be focusing about the concept of the human atlas. And what we want to do here has already been outlined by Katrin. I will try to show this a bit more from an infrastructure and technical perspective. So we are trying to build here a system powered by software and infrastructure that connects different facets of brain organization. And of course, we start with maps of the brain in terms of their regions, but the system is built to address the variability of the individual regions in the same system. Katrin has shown how this varies, for example, in the visual system. But from there, to be able to switch over to a different spatial scale down to the microscopic level to see in more detail how an individual brain region, what it looks like at a cellular level, and what the variance inside each region is. So we're trying to build a system where it can go all the way from the whole brain down to cellular detail, crossing the scales. And then there's another dimension to this. This is the dimension of different modalities or different principles of brain organization, not only cells, but fibers, receptors. Katrin has shown that. And the system is designed to build links also across those modalities. This can be in the form of connectivity data that has been computed for a given brain parcellation and shows the connection strength between different regions of such a parcellation. But this could also be measurements inside different brain regions, characterizing how, for example, the molecular architecture in a specific brain region is organized. You see here some example images of receptor densities. And such information can be linked to a brain structure, conceptually to the name of a brain structure. It can be linked to a space, to a coordinate of that space, or a range of coordinates. There are different ways to build such links. And we try to build a system that is very flexible in terms of these of such links. So this needs to become accessible and usable by many people. And since we are here including large data sets, especially when we go down to the cellular detail, what you see here on the right, we need to put that on an infrastructure. And that's what we have been doing in the last years in the frame of building the infrastructure EBRAINS. Here we have used data sharing services, a database in EBRAINS that's called the knowledge graph that describes the metadata of all those different contents of the atlas, the different maps, the different reference spaces, and the data sets that are linked. They are all fair data sets shared in EBRAINS. They have a DOI. They can be openly accessed. We are also using an infrastructure that is called Phoenix that provides virtual machines, storage, and of course also cluster computing services, which we use to host the data, to compute the brain maps, things like this. We are also building links to other repositories that provide structured interfaces, such as the Allen Brain Map or Neuromorpho, which is a quite well-known online repository, and others to make links that point even outside of our own framework here. All of this needs to become accessible, and we are trying to target here a range of different use cases, which means that we have to provide both interactive access, like a low threshold way of just exploring what we see here in a visual interactive way. That's what you see here on the left, We have, for example, implemented a 3D viewer that allows access to this framework. That's called Zebra Explorer. I will show that in a second. But also serving use cases that provide programmatic access and that allow users to build data analysis workflows based on atlases, or to connect atlases to their own software or simulation workflows. For this, we have a Python library that's called Zebra Python that allows full computational access, scripting access to the framework. I'm going away from my slides for a second to show you this interactive way. The first way, that's the Zebra Explorer. This is just an online viewer that is accessible for everybody on the web. And here you see this cytoarchitectonic atlas that Katrin was mentioning. And very briefly, in the interest of time, you can here click a brain region. You see the probabilistic map, and you see here a list of linked regional features. I'm here showing, for example, a receptor density in the selected brain region V1. Yeah, all of these things that we are seeing here are themselves data sets that you can find on the EBRAINS knowledge graph. They are linked from here, so you can, each element that you find here, you can also find it as a well-described, well-structured data set on EBRAINS and download the data independent of the Atlas ecosystem, of course. And as I said, we want to cross the scale. So in the same Atlas viewer, you can also select the big brain model, which is based on microscopy. And here you can now zoom in into much more detail. So this is a 20 micron model to which we are constantly linking data at even higher resolutions of 1 micrometer. And here you can see that I can zoom in here in the browser to the level of individual cells, while still having the 3D context of this whole brain available and having links to the 3 atlas in the system. I was here very brief in the interest of time. You can do the same thing with this Python library. I'm not going into detail here. This is openly accessible. You find a link here. But the idea is really that you can, with a few lines of code, do the same things and access parcellations, reference spaces, sample from the big brain at high resolution, and also find such regional data sets. Here you see also neurotransmitter receptors, cell body densities, and so on, which you would then programmatically access and have them in the form of common Python data types, such as pandas data frames, nifty images, common data structures. Now, all of this is not worth so much if it is not a living system to which we can integrate more data. And we are implementing several strategies to link new data continuously to this ecosystem. I'm only mentioning a few of them now. There's, for example, a tool that's called Quickly, developed in Oslo, that allows to anchor 2D histological sections to a brain atlas space. And we are developing a tool called Voluba, which allows to anchor high-resolution volumetric data to high-resolution templates such as the big brain. And I only want to briefly mention and show this. So this Voluba tool can be used if you have a tissue block from a histological experiment, such as this piece of tissue here. It can be used to upload it onto such a cloud service, and then interactively manipulate the position, the scale, also add landmarks to place it at an appropriate location in a reference space to give it more context, to clarify the 3D orientation, the size, the closeness, and spatial relationships to other structures in the Atlas ecosystem. From here, you can then, once you are there, you basically have access to the whole Atlas functionalities that I have shown. You can open this same data set then in the Atlas viewer, see information about close by regions. Here in that case, STS2, for example, has been discovered as being a close by region, and you can access all this information in the context of this data sets. And these are strategies that we are using to basically enrich the system continuously with more data and more measurements. This has also been used already by some researchers, also some who are here with us in the webinar, like Tim Salditt has beautiful data sets like this one, which we have started to integrate with the big brain model. Tim will certainly mention this as well. And with this, I will close my part of the presentation to pass over to the next speaker. Thank you.
Yes, hello. I'm Tim Salditt here from Göttingen, and thank you for having me and for the opportunity to share some of our results by phase contrast X-ray tomography on post-mortem mapping of the human brain. And if we talk about the landscapes of the human mind, irregardless of the level, anatomic level, cytoarchitecture, or maybe in future increasingly on the connectomic level, what you want, since landscapes are three-dimensional, you want three-dimensional imaging, and this is of course a challenge. And the classical paradigm is really based on sectioning, and whether for a basic neuroscience or for pathology, neuropathology, you can reconstruct three-dimensionality by adjacent sectioning, but you cannot do this in high throughput and not at isotropic resolution. And this is a very limiting factor if you wanted to really zoom in and out and approach really a multi-scale three-dimensional reconstruction. What if instead of that, you had the opportunity to go through sections just virtually from a whole data set? This is a movie that plays through a phase contrast reconstruction of 1 millimeter cross-section tissue, post-mortem paraffin-embedded tissue of human hippocampus. And here we see a higher resolution region of interest. You see, as we go through the dentate gyrus, on one side, we see very strongly contrasted plaques. So as you can guess, it's from an Alzheimer's patient. Here we have segmented the vasculature, and we can see how, in this case, mineralized plaques are positioned with respect to vasculature or the dentate gyrus or other regions and features of interest. And the nice thing about this modality is that it's non-destructive. You can do this on a volume, and afterwards you can still use the full capability of immunohistochemistry and conventional histology that you can do on sections to identify what contrast you have in your X-ray data set and with the information of specific proteins. On the left, I show you an example of recent work that we were able to do with in-house micro CT. So that's the topic today, and it really has two aspects: the atlas, the enhanced capability of having digital twins and representatives, but also this usefulness for clinical histopathology, where you know that essentially we're still working with the sectioning and optical microscopy that has been around for quite some time. As opposed to that, we now take a volume and we use X-ray imaging, but not by this contrast mechanism of attenuation, but simply by the property of matter that X-ray wave propagates at different speeds, and hence there's a phase contrast, phase delay of the X-ray wave at the exit of the object. And then these distorted wavefronts interfere and give you a measurable signal that still needs to be reconstructed. So this is a lot of the work that we have to do as physicists to really find the best way to calculate a sharp image. And if you wanted to do it at high resolution, nanoscale, let's say voxel sizes between 50 and 200 nanometer, you have to use a magnification scheme. And this is where X-ray optics and brilliant X-ray radiation comes in. We need large synchrotron sources. And for that purpose, as a university group, we've been able to work together with DAISY and put up an X-ray microscope for this type of imaging. When we started to work on this, that was all mouse. It's the ideal way to approach brain tissue imaging, neural imaging. We work with slices, different embeddings. We saw that you can get different contrast depending on whether you have it in PBS or in alcohol or in paraffin. And since the clinical material is to a large degree all stored and archived in formalin fixed paraffin embedded tissue blocks, this is what we then selected. So tissue is unstained, and we teamed up with Christine Stadelmann here at Neuropathology to, with this imaging capability, also tackle questions on a small cohort and ask, how can we, for a specific brain region like human hippocampus, see and monitor pathological changes in the cytoarchitecture in the context of neurodegeneration, for example, for the case of Alzheimer's disease, and as opposed to just imaging plugs or investigating a particular hypothesis, how about just measuring and then ask the data where you find changes and where changes come about? And this is what we set out to do. This was the project spearheaded by Marina Eckermann in her PhD. And of course, it all starts with the 2D map, conventional histology. Then we can select the region of interest. We can stitch tomogram by tomogram for an overview, let's say, of 8 millimeter cross-section. Pixel size is 650 nanometer in that modality. And we can go layer by layer and then locate things that we want to see and the structures that we want to represent and quantify. So here's a small movie that shows how we select the region of interest, dentate gyrus, and how we can reconstruct features. In this case, you see the segmentation of nuclear granules, the nuclei of granules. The holes in that structure is where the vasculature comes in. And we have the ability to zoom in further using divergent X-ray beams and then really look into certain features here, plugs again, or a particular nucleus, and find about the 3D distribution of densities in these structures. Then you can do this on many samples. Let's say 20 different samples, 10 from Alzheimer disease, from subjects that suffered from Alzheimer's disease and age-matched controls. And using machine learning, you can segment, for instance, in this case, the nuclei of the granules. And then if we analyze that without a prior class attribution, we can see that there was a major change from in this cohort, where you can see that these nuclei become more compact and more heterogeneous and more dense as we move along the main axis of change. And then if you switched on the neuropathological staging afterwards, you can see that there's really a pathway from, let's say, physiological to pathological structures. And if you want to interpret this, it looks as if this is a manifestation, in fact, of cellular senescence, the nuclei shutting up to a more compact state. So that's just an example how we can use these high throughput, large volume, high resolution scans, and then ask questions and evaluate that data. Now, towards higher resolution still, I have a first result here by my current student, Jakob Reichmann, where we were interested in Lewy body dementia and looked at a sample of substantia nigra. In the same spirit, we take first a larger overview, 1 millimeter cross section. We then zoom in into particular. It is not easy to find these Lewy bodies. And then we go at a resolution of 90 nanometer. Using extremely brilliant radiation at the European Synchrotron facility, beamline ID 16 at ESRF, and we get really what we think is superb data. So these are just examples. There's more on the way. We want to scale these methods to both to higher resolution and to larger brain regions that are meaningful and that could be inspected. And thank you for listening today. I want to credit my former PhD students, Marina Eckermann, now a postdoctoral scientist in Grenoble, Mareike Töpperwien, who has started neuroimaging in the group, and my current PhD student, Jakob Reichmann. And of course, none of this would be possible without this collaboration with my colleague, Christine Stadelmann, and her team in neuropathology. We also work together with colleagues from mathematics in trying to find morphometric parameters and to evaluate this very well. Thank you for your attention.
Thank you, Tim, and everybody. This is really fascinating data. I'm happy to share my screen. So thank you for the invite to present. This is collaborative work across many different consortia and also including HubMap, the ambition to map the human body at single cell resolution. It's my great pleasure to present here in this forum on the Human Reference Atlas. Some of you might be part of this effort. Some of you might be new to it. What we would like to accomplish is to create a 3D reference space that can be used to map major anatomical structures and cell types, ultimately also identified by characterizing biomarkers. All of this should be linked to existing ontologies, and we are in the process of extending existing ontologies so that they properly capture healthy human structures across organ systems. Second, we would like to have a mapping that helps us map new data to this reference atlas, and Timo already showed examples for that. And we would like to have an atlas that's authoritative, that's computable, so you can run API queries against. It's not just a book, an atlas books that you hold in your hands. It should be published as linked open data so that it connects well with other data sets, for instance, on diseases, on food items. It should be open so that anyone can use it for research and teaching and clinical practice. And it will be continuously evolving, as almost every other atlas is. And so there is a paper which is linked from here where a number of consortia are now together in this, because it will take a village to build such an atlas. And the different teams really focus also on different organs. And you have here the list of all the organs that were in focus as of last year. I think there are a few new ones in the mix now. And you see that the brain is one of the highly studied organs and also one that enjoys quite a bit of funding. So it's wonderful to see what can be done for the brain. But keep in mind that every organ is actually quite different and has different opportunities for creating an atlas and a mapping process to map new data to it. You might know our work from mapping science, technology, innovation, job market landscapes, educational landscapes. So we have created a number of atlases and have an excellent exhibits that goes around the globe. Again, some of you might have encountered it at libraries, at science museums, at national academies. And we can learn much from the history of making maps of cartographic spaces, but also of abstract spaces such as collaboration networks or science itself. In general, it typically takes quite a bit of time. Here on the right-hand side, you see a map of India, and it took generations, literally generations, to create this network of repeating sightline triangles that was ultimately used to measure the space and extent of India and give it the true shape. We hope it doesn't take decades to get to an atlas, because we all believe that an atlas would be very, very useful for understanding how the human body works, but also helping us cross-search across different efforts that are now all mapping the human body at single-cell resolution. Just as first maps of our world, here a map from less than 300 years ago, were not perfect. The initial maps of our human body will not be perfect or complete. They will focus on major organs, and I think brain is one major organ that is the focus typically of this talk series. In order to map something systematically across organs, across labs, it is important to have a system in place that helps unify data. In the Human Reference Atlas effort, we look at anatomical structures that are nested or branching. We look at cell types that are commonly located in these anatomical structures. And we also record characterizing biomarker sets. And we connect all of this to 3D references at the organ level, as you saw earlier, in terms of brain, but also at the functional tissue unit level and at the single cell level. These so-called anatomical structure cell types biomarker tables can be mapped and visualized so that you can interactively explore them. And here you see a network for the spleen, and you see the corresponding 3D spleen organ and white pulp of spleen, one of the functional tissue units, but then also biomarkers, genes, proteins, lipids, metabolites that are characterizing certain cell types inside of certain anatomical structures. And many of you might also use cell type annotation tools such as Asimus or Cell Typist or Prof to annotate genetic data sets that are now available at the single cell level. But using these annotation standards helps unify cell type annotation across different efforts. And of course, there's also one for the human brain and also for mouse brain, the motor cortex especially. Within HubMap, we have been developing so-called OMAPs, validated antibody mapping panels. And here you see one for the skin aligned with the AST+B for the skin. And then here you see the affinity reagents for different types of cells. And there's a lot more information about OMAPs behind this link. And I think all these slides will be shared. Jeremy Miller last week presented in the AST+B working group a new extension of the AST+B table for the human brain. You might like to check that one out. There's a lot of major improvements from the previous digital objects that we had for the human brain. And we also now have first registrations into the 3D human brain that actually is adopted from the Allen 3D brain for the male and female human body. And here, data from a very specific study from the Allen Institute were registered in 3D. Timo also just showed how you can use the Jülich Brain Atlas to map new data. And we are very interested to create projections from the Jülich Atlas to the Allen brain. For Atlas construction, it takes very detailed, careful data collection and processing, ideally using entire organs, which is not always feasible. But here you see our millitome process for using a reference organ to construct a 3D cutting device to ultimately cut entire organs in 100 or 200 pieces and to then register them inside of the 3D reference organs. And these are the instruments you would use. This is the butterflying of the kidney. This is how the tissue blocks are preserved and frozen. And this is how they would be registered, typically one by hand. But using the millitome, they all would be registered semi-automatically. Happy to present more information. We now have collision events with more than 1,500 anatomical structures, which then automatically serve as anatomical structure tags for the tissue blocks so that you can retrieve them based on these tags. You can then explore all these anatomical structures and cell types and registered tissue blocks via the exploration user interface. And I think there are links now in the chat that you can play with these tools yourself. And you can then zoom into certain areas. You can explore different types of data that are associated with these tissue blocks. And you can also run spatial search. You can click on a data block and you can see all similar, spatially similar tissue blocks. But you can also take this golden sphere to identify a volume that is of particular interest to you and then retrieve all data and download it for offload computation. You can also use the Vitesse tissue browser to see details on different types of assays. We have more than 30 assay types within HubMap. And you can also, of course, explore UMAP or TSNE plots of cell type populations generated via the gene by cell matrices or protein by cell matrices. We are in the process of implementing interactive user interfaces for functional tissue units so that you can explore what cell type populations exist for which populations, but also what gene, protein, and lipid biomarkers are common and what expression levels are common for different sex, ethnicity, and age groups. And here is the set of the initial 19 functional tissue units we are focusing on. None for the brain, maybe in the future. This is more information on how we actually get the data which is specific to functional tissue units, also using Kaggle competitions and engaging thousands of teams from around the globe. And even though the initial HubMap and ultimate human reference atlas will not focus on all organs, it will be multi-scale, really helping you to zoom into an organ, to a functional tissue unit, to cells and their interactions, down to the protein biomarker level. And in some very exciting collaborations, we have been able to show how that might work for skin here, for instance, where you register specific tissue blocks, reconstruct tissue in 3D, and then also start to look at using the vasculature as a latitude-longitude system for the human body. And I would be happy to answer questions. And also there is a lot of training and outreach that is available for HubMap and the Human Reference Atlas. It's part of the Atlas portal. It's part of the working group, which you might like to join. It's every first Wednesday of the month. The next one is on April 5. And there are many standard operating procedures now on how to build this atlas and how to use it properly. And there is a visible human MOOC that is free for anyone to enjoy. We also have 24-hour events, which you might like to check out. And I think we will host another one, not this year, but in December 2024. Thank you.
Okay, fascinating. So let me share my screen to sort of get going. Okay, so basically sort of we're changing gear a little bit. So as Katrin already pointed out in her presentation, we are now going to research that basically uses these atlases and uses these ideas to sort of put functions of the human brain into a space. So what I would like to do is to take you on a journey through the human pain system. And this is a very, let me get my pointer. This is a very old indication of a certain idea of a system that sort of from the skin efferents go through a tube into the brain from René Descartes many years ago, but actually not that bad. If you look at the sort of more contemporary atlases, we have a brain system where you have peripheral efferents entering the spinal cord and the dorsal horn, crossover, branching off at various stages in the brainstem or midbrain, then basically synapse in the thalamus, and then project onto cortical targets. This is the classical pain system. What is less known is that we have a so-called descending pain modulatory system, and I want to talk a little bit about why we have that and how it works. Coming from cortical midline areas, the cingulate cortex, but also from the insula, having a major hub in the midbrain, the periaqueductal gray, then projecting over the rostroventromedial medulla onto the dorsal horn of the spinal cord to exert its modulatory influence on the pain that's coming in here. So why is this important and how does this work? So we have investigated together with Ulrike Bingel, whom you see here, who was a postdoc in the lab many years ago, the idea that expectation can change how you feel pain. Sometimes this is also known as placebo analgesia. And in the first functional imaging studies, we could identify that we get an activation increase in the rostro-anterior cingulate cortex and an increased coupling to a PAG. This is actually a very old slide, and you've seen much more beautiful atlas slides, and I probably should change this. So, but then we asked the question, how is this mediated? How does the brain actually sort of downregulate? So we basically wanted to know the sort of, well, how is this implemented? And the idea was that it's probably endogenous opioids, endorphins, that are released by expectation and thus get you a decrease in pain perception. So what we did is we treated one group with basically saline, and another group was actually treated with naloxone. And as you can see here, this difference in the saline group is the placebo analgesia effect. And it's much smaller if you block endogenous opioids, basically showing you that the effect is mediated by the release of endorphins. And this is basically showing you that the activation in the rostral-anterior cingulate cortex is also diminished. And importantly, this also involves opioidergic structures like the periaqueductal gray. Furthermore, the connectivity between the rostral-anterior cingulate cortex and the PAG that we showed to be sort of elevated in placebo analgesia, sort of a network effect, is actually blunted in the group where we have used this very selective mu-opioid antagonist naloxone. So telling you that it's the effects of endogenous opioids that mediate the analgesia by expectations. Now we've taken this further because, as I've shown you in the very first slide, that pain is mediated or basically sort of also happening at the spinal cord. So we have evolved fMRI to also do imaging of the spinal cord. So this is a human spinal cord. And many studies that we've done in our lab actually show very precisely a location of the dorsal horn activation in the spinal cord. And we've also taken back the idea that maybe in placebo analgesia by expectation, this already happens at the first synapse of the central nervous system, namely here at the spinal cord. And Falk Eipert, who did this study, could actually show that the activation to a placebo-treated painful stimulus is very reduced as compared to a stimulus that has the same intensity, and basically showing that a modulation of this already happens at the first synapses of the central nervous system in the spinal cord. But we also wanted to look at the interaction between the brain and the spinal cord. So we were very lucky that our physicists could develop a sequence where you can image the brain and the spinal cord at the same time. And Christian Sprenger did the first study with this sequence. So here you see main activations of pain that we've seen before and main activations of the dorsal horn spinal cord. But more importantly, he could show that the connectivity, the coupling between the PAG and the spinal cord is nicely correlated with perceived intensity of the same temperature. So the temperature was 48 degrees, and the rating of how much you perceive in pain was correlated with the activation or the coupling between the PAG and the spinal cord. So in the final part that I want to share with you is that instead of making things nicer by expectation, placebo, we also unfortunately have the opposite, that things go worse by expectation, and that's called the nocebo effect. And it's often basically accompanied by drugs where you think you have a side effect, although that side effect is not coming from the drug. So Alexandra Timmermann, who was a PhD in the lab, created two creams. They were invented, they don't exist. And basically, she told people that they were against itch with neurodermatitis. And the two creams, one was called to be more expensive than another one, which was confirmed by a behavioral study. And we actually wanted to see whether these creams can, by expectation, increase pain. That's what we told them. It's good against itch, but it unfortunately gives you a little bit more pain. And this works. So you see this more expensive cream gives you more side effects than the cheap cream. This leads to activation of the periaqueductal gray. It also leads to deactivation of the rostral-anterior cingulate cortex that actually scales with the individual effect of how many side effects you get. That means how much pain you get by this cream. And this is sort of basically also shown in the spinal cord. We've done another study to look at that, very similar location. And to finish off my talk, I want to show you why we are so keen on looking at spinal imaging and brain imaging at the same time, because we could show that the coupling between the spinal cord and the PAG and the cortex basically can nicely dissociate between the two effects of side effects, namely the cheap and the extensive cream, or a sort of bigger side effect, more pain as compared to the other one. And I'm very sorry for the technical glitch. I hope you can still, or you still have heard most of my presentation, and also as everybody else, I'm happy to take questions.
Thank you very much, Christian. This was really an exciting journey through the pain system, and I think you are one really of a few people that I know that is taking care about activations in the spinal cord. And look how precisely you can image and visualize it. So we have received already some questions, but we could have more. So go to the F&A and place your questions to the F&A section. And I would simply start to read from the very beginning. And the first question was, are these functional connections based on task-based fMRI or on resting states? This was related to my presentation. And indeed, these data come from the 1,000 brain studies, and we have many data in it, but this was a resting state fMRI. There are diffusion data as well, and also task-related data. Everything is, so to say, in the atlas. And then Didem asked, is a large area delineating the dorsolateral prefrontal in the segmented 3D brain shown actually a single monolithic area cytoarchitectonically, or is it just different from other areas and is going to be segmented further into sub-areas? So what I was showing, these were maps of 3 premotor areas. So some researchers would not include it into the dorsolateral prefrontal cortex in a strict sense. So this was premotor cortex, only the ventral part, and we see 3 areas. There are 3 more areas in the dorsal part of the premotor cortex, and there are also 3 areas, I would say, in the medial part. When you think about the whole prefrontal cortex or lateral prefrontal cortex, we have an estimate of a few dozens of different cytoarchitectonic areas. Then, Timo, we have a question to you. Is this framework going to be extended in the future? In which respect and by whom?
Yeah. Oh, that's a that's a good question. So yes, it is designed from the ground up to be extended, and there are maybe just different levels how people would contribute. So of course we are working with labs and inviting labs to contribute data. There are curation teams that help to link more data into the system, share your data with this, but people can also extend in terms of functionality. For example, the Atlas viewer provides a plug-in architecture where people could write functionalities that are integrated into this interactive system, and there are structured ways of also adding functionality on the Python side to these systems. So yes, it is fully thought to be extended in terms of functionality and data.
Thank you. Tim, we have a question for you. To reconstruct neuronal circuits, tremendous efforts are undertaken by electron microscopy. Do you think X-ray tomography can reach the required resolution?
This is an excellent question, and I think it's too early to answer that. What I showed you today was really unstained tissue, and that also means that there's a tremendous dimension now to really exploit radio contrast agents optimized for phase contrast tomography, rather than what happens now occasionally that we take protocols from EM and say, okay, that's probably going to give us a boost in contrast, while it must not necessarily, it can also be overly contrasted. Once that we understand how to correctly label larger 3D volumes, I think there's just based on the optics and the photons and the information that is in the data, potentially, I think there's good reason to believe that we can scale the method and hopefully go to the synaptic level. And then, since we would probably have a completely different throughput capability still, I think longer projections could be traced. Yeah, probably the image quality will always lag behind EM, but if it's good enough to trace larger projections and see synapses, that would be great. Not shown yet, it's speculation, but it's definitely something that we and others must and will try.
Thank you, Tim. Now 2 questions to you, Katie. So you have an OFN. How can I contribute to the Human Reference Atlas? Perhaps you can share a link. And perhaps also the second question, Katie, India mapping, very true. GTS took 55 years through planned for 5 years. Brain mapping, what sort of time frame is expected? So if you can comment.
Yeah, so I can put the link to the working group again. So if you have your own 3D ideally atlas, also if it's just a 1D atlas. So for instance, for the gut, we actually have a one-dimensional atlas, which is very valuable for understanding how deep in you are into the colon. So dimensionality is different for different organs. But if you have a 3D reference atlas, for instance, for the brain, please do come join us for the working group. The link is in the chat again, and find out how you can project from your atlas to our human reference atlas, because then you automatically can search across 26 plus organs, and you can identify what cell types are commonly found in which organ and where. And ultimately, there are also many experts interested to ask for certain biomarker expression values for, again, genes, proteins, lipids, metabolites across organs. I think we are all in this together. The organs are in this together. They cannot exist on their own. They depend on each other. And I think we want to have a more holistic understanding of the human body. So that's maybe for that part. So please come in and also look at the work that has already been done for the brain. We are always watching out for reviewers. And I think there might be quite a few experts on the brain in this session, and they would be very welcome to serve as reviewers for the existing human reference atlas for the brain. The next question was timescales.
Yeah.
It took a long time to map the human genome, and that was a one-dimensional structure that we all pretty much share. There's not as much human diversity for the human genome as there is for our human bodies. And the human body is very much a 3D system. Each cell exists in a 3D environment, and it matters what it experienced to get there, right? From the initial one cell to the 37 trillion cells. So we have to understand that better. And I don't have a good guesstimate, but I believe if we all work and collaborate together, it will be way faster than if we try to do this in parallel. And that's really what the Human Reference Atlas is trying to do, to bring good experts together to help each other understand how to do this best across organs, across scales.
Yeah, I think we cannot more agree, because having now built an infrastructure or an informatics system, yeah, where these things can be included, and this is the same for Timo's work, now it can go much faster than when we started, and nothing was there to build. Christian, for you now, the effect of cheap versus expensive medication should also be related to the basal orbitofrontal cortex as well. Have you investigated their relationship in terms of similar regressions with pain perception?
That's a good question, Didem. So actually, we looked at this area across different tasks, placebo and nocebo, and we found there is basically a one-dimensional mapping. So we would even argue that this area would map the sort of value of treatment, where in placebo, you see an increase, it's sort of a high activation, whereas in nocebo, you get basically a negative activation. So the better your treatment or the value of treatment, the more activation you get, and that nicely aligns with all other decision-making tasks that look at this area.
Okay, thank you, Christian. So I would say there's time for last question. We are approaching the end of the seminar, are already a little bit over, but if there are burning questions, we would of course address it. Nothing burning, I would say. So thank you very much, all the speakers, for making this seminar possible. And thank you also very much to the team behind from the Human Brain Project. And for you who were joining our webinar, have questions. So it's great that you are so curious. If you have any question that come into your head after this meeting, no problem, just contact us. And having said that, I thank you again for participation and wish you a very nice evening, and hope to see you perhaps at the summit of the Human Brain Project, which takes place in the last week of March. Come and join us, or at the next webinar that we will have here very soon. See you and have a nice week.